Good morning, everyone. Bowing to the lotus feet of His Holiness Jagat Guru Dr. Sri Shivratri Rajendra Swamiji and seeking the blessings of His Holiness Jagat Guru Sri Shivratri Deshikendra Swamiji, we begin the program. Friends, we are virtually assembled here for a webinar on career opportunities for life sciences graduates in pharma and biotech sectors on the occasion of 105th Jayanti celebrations of His Holiness Jagat Guru Dr. Sri Shivratri Rajendra Swamiji and also commemorating the International Microorganisms Day. I, Dr. Ashwini, Assistant Professor and Coordinator, Department of Microbiology, take this opportunity to welcome all the dignitaries. I thank our beloved Pro-Chancellor Dr. B. Suresh, Vice-Chancellor Dr. Surinder Singh, Registrar Dr. Manjunatha, Director Academics Dr. Kushalappa, and Dean Director Research Dr. S. Balasubramaniam for sending their best wishes for the success of this program. I welcome our HOD, Professor K. Ravisha, for this event and thank him for accepting our invitation and giving us constant support. And most importantly, I wholeheartedly welcome our today's speaker, Mr. Vishwarup Das Gupta, for this program. Welcome you, sir. I also welcome all the faculty from JSS and other institutions to this program. And last but not the least, I welcome all my dear students for this wonderful program. I now request Professor K. Ravisha, HOD, Department of Water and Health, to give the opening remarks.
Hi, Mysuru. This is Ravish here. Namaste.
Namaste. How are you, sir? After a long time.
Fine, doing fine. Very happy to see you on this occasion, that too at a time when the Department of Microbiology has been newly started. And this is the first program under the aegis of the Department of Microbiology that we are doing it. And I'm also happy to invite you. Thank you very much for accepting our invitation to deliver this lecture on the International Microorganism Day. My pronouns to the holy feet of His Holiness Sri Rajendra Swamiji, the commemorating his Jayanti celebrations. This program is being organized. He is a man known for very specific activities, one of the important activities being Jnana. I also invoke the blessings of His Holiness Sri Deshikendra Swamiji. A very good morning to all the participants. And good morning to the speaker, Mr. Vishwarup Das Gupta, who has accepted our invitation. I thank him for having accepted our invitation at a very short notice and being available to deliver this lecture. Friends, as you all know, the International Microorganism Day is relatively new. I think if I remember right, it started sometime during 2017 and onwards. It is celebrated on 17 September, the day in 1683 when the Dutch merchant Anton van Leeuwenhoek, with no formal education or university degrees, first described a single-celled organism and communicated through a letter to the Royal Society of London. Thus, he reported for the first time the existence of a single-celled organism, which nowadays we call it as microorganism, as it is not visible to the naked eye. IMD was established by the Portuguese Society of Microbiology, but of late it is observed worldwide. There are also sayings that even UNESCO is trying to adopt this. The purpose of the celebration of this day is to raise awareness about the importance of microbes among society in general and among younger generations in particular. The celebration of this day is much more relevant in the present day pandemic situation due to severe acute respiratory syndrome coronavirus, widely known as SARS-CoV-2. Anton van Leeuwenhoek was a tireless worker, gifted with an extraordinary power of observation, displayed infinite curiosity. He designed and developed his own crude microscopes, perfecting the lenses of his optical system, and thus was successful in obtaining extraordinary applications. Well ahead of his time to observe and describe for the first time microorganisms. In his famous letter to the Royal Society of London, an exquisite description was made of the first observation of living bacteria present on the dental plaque, which was accompanied by drawings of microorganisms and their movements. Thus, he laid the foundations for microbiology as a discipline of study. It is now necessary that International Microorganism Day is celebrated in several parts of our country to popularize the art of living with microbes and loving microbes, instead of only hating them for their disease-causing potential. Many activities could be conducted for the benefit of society, particularly to showcase the activities done by the scientists of our country in the field of microbiology, open laboratories, do-it-yourself experiments or exhibits, microbial product testing, hands-on training through workshops for high school and undergraduate students, and workshops for high school teachers. Similarly, an open day on an International Microbiology Day by all the microbiology departments, be it medical microbiology, agriculture microbiology, industrial microbiology, or various other agencies or institutes that are working, would give a very good boost for understanding microbes per se. I have one thing to say just here, because as we all know, when an enemy comes, a macroscopic enemy comes with a dagger, our enemy comes with a dagger, the opponent will be very careful and immediately he reacts. But unfortunately, if you see the present situation in the society, microbes being not visible to the naked eye, however much we are trying to educate the society to take care of themselves, saying that this could be an important enemy for you, it can attack you, they are not unable, the society per se, the common man is unable to actually visualize it, because of which the number of cases is continuously increasing in the world. So I wish people become aware of these things and they say themselves, I hope the Almighty will give all the strength to the general public for by boosting the immunity to save themselves in this crisis hour of national pandemic, in fact, a worldwide pandemic. I thank Mysuru again thank you for accepting our invitation and delivering this inaugural address. Thank you.
Thank you, Ravisha sir, for enlightening us about the importance of International Microorganisms Day and also about microorganisms and their importance in our society. Now let me introduce today's speaker. Mr. Vishwarup Das Gupta is presently working as Director Quality Assurance Gene Therapy in Serpentina Therapeutics, USA. Mr. Vishwarup was born in Chittagong, Bangladesh, and he has his education roots in India. He did his BSc from Bangalore University and pursued his master's in microbiology from the University of Mysuru and graduated in the year 1999. He has enormous experience as quality and compliance leader for over 18 years, leading across functional teams in manufacturing, quality control, and quality assurance. With this crucial industry experience in pharma and biotech sectors, he has acquired core professional strengths of development, remediation, and implementation of quality systems, isolated technologies, aseptic processing, contamination control and compliance, training and mentorship, internal audits method, equipment validation, and quality risk management, and the list goes on. Mr. Vishwarup started his career in Bangladesh in 1999 in Opsonin Pharma as a microbiologist and later joined GlaxoSmithKline as Quality Assurance Officer in 2003. With this crucial initial experience in pharma sectors, he later moved on to USA in 2004 as microbiologist at Atlanta Pharma and was responsible for operation and maintenance of the industry in clean room and environmental monitoring. In fact, he was awarded with Atlanta Pharma Annual Award for his idea implementation that helped the industry to save the huge money. Later, he moved on to GlaxoSmithKline Biologicals and worked in various capacities as microbiologist in QC in Marietta, Pennsylvania, and QC microbiology supervisor. And he was also responsible for setting up the microbiology laboratory and established the material and personal flow in the lab at the startup site. He was elevated to the post of Deputy Director Quality in Rockwell, Maryland, where he prepared and led the successful pre-approval inspection at this role in 2011. He has served in Sanfoy for nearly a decade and had responsibilities in various capacities as manager, sterile assurance, Rockwell, Maryland, deputy director, associate director, global product quality, associate director, compliance, Massachusetts, and quality leader. Mr. Bishwaroop, at present, working in Serpenta Therapeutics as Director Quality Assurance, Gene Therapy, Andover, Massachusetts. Managing a team responsible for providing quality oversight of cyclic GMP activities at contract manufacturing organizations and service providers used to manufacture clinical or commercial product. So leading multifunctional team and works within the quality organization to ensure the appropriate scientific, regulatory, and quality standards that are followed to assure product and quality and complaints. With this brief introduction, I now request Mr. Bishwaroop Dasgupta to address our virtual gathering.
Ashwini, thank you so much. First of all, that was really a quite humbling introduction. And first of all, I really want to thank you for giving me this opportunity. After that, obviously, being very honored and privileged to be here today, being a student of Mysuru University, Department of Microbiology, Dr. Avisha, obviously, respected sir, thank you so much again. Nice to see you after such a long time. I would like to also thank Dr. Suresh, Dr. Surinder, and obviously the panels there from GSS faculty, again, to give me this opportunity. Good morning to everyone. It is it is morning here, 12:42. So I can say that it is a good morning for all of us. With that, I will directly go to the presentation. I don't know how this is going to be managed. If it is interactive, really, that will be really fine. Ask me a question anytime if you have any question, or you can hold it at the end of the session, and I will try to answer as far as my knowledge goes. So with that, the reason I thought this is very important because when we were students, it was so much academic that we were just focused on the books, right? As Dr. Avisha was saying, that we studied all the soil microbiology, aeromicrobiology, medical microbiology, industrial microbiology, whatnot. But I don't know how much we gathered experience in terms of industry, that when we come out, what we should expect out there, right? Many of my friends, Dr. Nagendra Prasad, and many others, I know that they pursued in this field, but people like us, some of us chose the industry to move on because there are a lot of opportunities, that's right. And I never thought that I can become a good teacher, or I can teach students, because I don't have that capability. So with this opportunity, then I thought that maybe let me present some of the sectors where I worked so that you know. I think it's a good opportunity. Maybe next year, I don't know, Ashwini, if you're thinking, bring people from other industry or other sectors who can give students some perspective that what they should expect when they come out. With that introduction, let me see if I can present my screen. Please let me know if you can see my screen. Ashwini, can you see my screen?
No, sir. You can share the screen over there below. You have an option there, share screen. Yeah.
Now you can see?
Yes.
Okay. Yeah. Something about me. This is my family. I think the photographer chose to cut me off because I was looking somewhere else and not towards her. So you don't see me. My son, towards the left, my older son Ayush, my younger one Shomil, 16 and 11 eventually. this year, December, he will be in, and next year he will be 11, my younger son, Shomil. And behind there, my beautiful wife, Subarna. I obviously, as Ashwini said, I graduated from Mysuru University in 1999, moved to different biotechs and in different sectors, and then started my career in the microbiology, and then moved to the quality organizations. And I will give you some idea about the differences. If I'm going fast, Ashwini, please feel free to pause me, okay? Sometimes I go fast, okay? So industrial microbiology, again, it is a vast sector. I'm just focusing on a very small part of the industrial microbiology. Again, only pharma and biotech. And also in the pharma and biotech, I will just focus on a little bit on the gene therapy part, not the entire pharma sector, because again, the time is not going to cover everything. And I don't have the knowledge in that sense to present all the entire sector. When we come to the biotech or pharma, again, I'm trying to generalize most of these things. People can challenge me and I will accept that. But this is basically whenever you're working in a pharma or biotech, it starts with research and development. Then it goes to the clinical side where they do certain clinical studies. And then there's manufacturing, then comes the quality, regulatory affairs, and it goes on. And there are supply chains who are responsible to manage the supply to the market. When it comes to the R&D, or some people call it product development, there is a lab. Again, I'm trying to make it very general so that people can understand, especially for the students, maybe it will be difficult. So that's why. So there is a lab who are responsible for, so say that a big guy came up with an idea that, hey, why don't we make this drug, right? So they come to this lab, they do some studies, they try to do a small lab experiment, they try to synthesize, and then they do different kind of animal studies in that lab. From there, they go to the pilot, little bit. Pilot means they just scale it up a little bit, and then they make the product again. They again test it out. They do the quality control, and then they transfer it to the next department. And these all are happening basically within the R&D or the product development. That's why they call it product development, because they're still developing this. At this phase, it is very important that they characterize the drug, they understand the safety profile of the drug, and they understand that what can be the side effect of this drug to the patient. They do a lot of animal studies. They try to do the toxicology profile, and then they try to come up with the dosing. So from there, as I'm saying, that they move to the clinical studies. Now, why I'm telling you this, guys, because again, just to give a broader understanding of the industry, and you can work in any of this. To work in the R&D and or in the PD, you need to have a very strong understanding of the science, so that again you can analyze the data. Along with the microbiology, we have seen that there is a great need in this place for the people who are statistician. And I mean, no one should get offended, but I've not seen many people in US who are born in US are very good in maths or statistics. Most of these people generally we hire, they're either coming from China, India, or some of the Asian countries. So I don't know in the academia right now whether we have introduced that analytical part, the statistical part, so that people can understand how to apply those. So that's very important. That's very important, I would say, to gain that knowledge. So before going to the clinical study, there are preclinical studies, as I said, from the lab. They started identifying the dose, the route of administration, patient eligibility, and the toxicity. The toxicity is very important because we don't want to introduce a drug that is going to be very harmful or toxic to the patient, right? So after that, the studies are done. Someone says that, yep, it is looking good. Let's move to the clinical trial. And in the clinical trial, we have multiple phases. And the phases are depending on the number of population and what we are going to test, use as a test population, or you can say test, let me say population because it includes animal, human also. So in this, again, we determined the study population, as I said. We designed a study that, okay, how we are going to introduce the drug, again, how we are going to select the dose and safety, and what should be the endpoint. Because we cannot keep on testing the product, right? So endpoint is very important. And phase 1, phase 2, phase 3, at some point, this is again by the population number and how you're taking it ultimately to the market is the goal. So at the phase 3, which is we call pre-commercial, is the phase where we try to use the drug in the largest number of population, gather the data, analyze, present it to FDA, and then try to get an approval to go to the market. Okay. So I don't know whether still you can hear me. So sometimes give me a nudge that again, if I'm going fast or if you're not hearing me, just let me know, okay? I can only see myself. That's why Ashwini is gone somewhere.
No, I'm here only.
Okay.
We are hearing you, sir.
It's going comfortable, okay. Nothing to worry.
Thank you, sir. Okay. Now comes the critical part, the manufacturing. And in this slides, I'm going to go a little slow and try to put some emphasis on microbiology, why that is important. So once all those phases are done, then we move the drug from the clinic, I'll say from the clinical phase to the commercial phase. And these all are like scaling up. Say that in the beginning, maybe we were making 20 vials, 100 vials of drugs, and now we are making thousands, right? So typically when we were young or in the college, we only knew drugs or pharmaceuticals, drugs like, I don't know, aspirin or paracetamol, those kind of drugs that are over the counter or the doctor used to prescribe us, right? These are called, or eventually we came to know that these are called small molecules. These are mostly chemically synthesized. There are obviously some exceptions, and we very well known these drugs, right? The chemical structures are very well known. So these are very well understood, the drugs. So these conventional drugs. And different forms, you see the tablets and all these are solid dosage, liquid dosage, obviously you get in the bottle, the ointments, the topical ointments that you get in the tube, apply on they are called topicals because you put it on the skin, or they're like, they are not injected. So that's why they are called topical. And then parenteral drugs are injectables. Parenteral means beside, that goes directly in the blood. So that's why they're called parenterons, not in the intestine. Enterons mean intestine. So these are injectable drugs. So that is your conventional. Then comes to the biologics. And I didn't have any experience in the biologics because I don't know in the beginning that there is a sector, huge sector that are dedicated to develop these vaccines or recombinant proteins or the cell or tissue therapies or the gene therapies. It's a huge booming sectors now. And where I live in US, Massachusetts, it is the Boston area. It is called the hub of biologics. There are like tons of companies who are working, developing this kind of biologics drugs. You won't believe in every corner, maybe there is a company, we call it startups. They are trying to come up with some ideas. These are the students or the professors who are coming out of maybe Harvard, MIT, or any other places. They come here, they put together a small lab, they do the experience, and they come up with an idea. Some of them obviously don't have enough money, so they sell it to the big companies, and some of them eventually take it to the commercial phase. The company right now I'm working, these are also startups. Sarepta Therapeutics, they are gene therapy company. Again, this is right now the booming market. Everyone is trying to put their hand in this because this is people are thinking the future. Because you don't want to keep on taking the drugs. If there is something that you can take one time and you're done with it, disease, then that is the goal, right? That is the dream. So those are coming to the large molecules, because these are now dealing with complex molecules that either protein or complex proteins that are going inside your body. And if you want to compare, like say that if a conventional drug, say if it is a bicycle, I'm just trying to make things up as a comparison, the biologics or biological molecules are like a jet plane, right? So these are huge molecules. And that's why they are complex to make. They're difficult to make. There are a lot of, these are very heat sensitive. They are prone to microbial contamination. So lots of controls are needed. Okay. So that is manufacturing. I'm just trying to give a very high level, broad ideas. And if hold your questions, if you have any, I'll see whatever I know, I'll try to answer. And if I don't know, obviously I don't know many of the things, I'll try to get those answers to you, okay? This is just, again, a basic, very high level basic idea. So that you start with a very small stock culture in the biologics and you scale it up. Then you try to harvest the material, then you try to purify the material, then it goes to your final product. Now, I put here a term, media fill. When you come to the industry, microbiology is of paramount importance in this industry. Microbiologists, people are looking for microbiologists. And I don't know, maybe some of you have exposure to different regulations. Obviously, Dr. Suresh, I know that he has been involved with some of the pharmacopoeias in US, like USPs and all. Maybe some of you obviously have heard this term. So when we make a drug, ultimately we need to make sure that it is safe for the patient. We cannot allow a drug to go in the market knowing that this is not safe for the patient. The media fill is performed to check the entire process with a media. Basically, we use the TSB. I think obviously all of you know what is TSB, tryptic soy broth, and that media is passed through the entire process. Whatever you're using, whatever equipment you're using, the hose you're using, it is passed through that, and ultimately it is filled to the vial. And these vials are then incubated to the incubators. There are two different temperatures that are used. For the bacteria, we use 30-35 degrees Celsius, and then for the fungi we use 20-25 degrees Celsius. So these are incubated for a particular period of time, generally 14 days. And after that, we check if there is any growth. If any, so there are some guidelines around there that how many vials can be contaminated, and you have to do a huge investigation to figure out why there is. But anyhow, regardless of that, we check that if there are any contaminated vials, if there is any, then we'll say, well, there is some problem there in our process. Because here what we are trying to make is a sterile drug, right? And if there is a contaminated vials, means what? There are microbes present. And this is where the microbiologist comes into play. Because if you put someone else who has no idea about microbiology, he or she may not understand that growth, right? They may see the turbidity and just throw it away because they have no clue what the heck they're looking at. So that's why in the industry, the microbiologists are very, 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 very important in our places. And we really recruit not only in the lab setup, also in the manufacturing. And again, as I was saying, if you look, not you, but whoever works in the industry, they know Annex 1, which is the European regulation right now revised heavily to put emphasis on contamination control. And whenever it is coming to contamination control, it is the microbiologist, right? Because no one understands this better than us. We know the environment. We know what we can expect in the environment. We know what is bacteria. We know what is fungi. Some other people who have no clue, they won't understand why this exists, where this exists. And during the hiring process, when we hire people, they go through rigorous training. And part of the training obviously involves, um, we call the people who work in this aseptic core, aseptic operators training, and we train them heavily on microbiology. Look, we spent 6, 7, 8, 10 years in the academia to learn, or in the college to learn microbiology, and we have to teach these people in 2, 3, 4 days about basic microbiology. So you can understand the challenge and how much they can learn. So that's why it is important that if someone is coming with a microbiology background, it is very, very helpful. Okay. So with here, so that's what so I say, as I said, the media field is very, very important, because again, the media to choose that you're using, a microbiologist can understand why I'm using TSB, right? Because it is a universal media. It is going to support, I know that, bacteria and fungi at the same time. On the other hand, if I choose there's no other liquid media that I can think of right now. SD is solid, but something that is not going to, again, support all the broad organisms. As a microbiologist, I'm not going to choose that, right? So, so that's why it is very important, again, a microbiologist be present in that group. As I'm in the gene therapy manufacturing right now, I just wanted to present this to you guys. This is a very, very interesting sector right now. Again, as I said, it is hot. All the big industries are trying to put their hand there. They're trying to buy the small companies with billions of dollars, because this is what future is, okay? At Sarepta, again, there are a lot of things that I cannot share because they are confidential and they are proprietary too. So that's why I couldn't put them on the presentation. It's not as simple as it looks like here, very complex process. But again, if you have any question, I will try to answer. Some of the questions I may have to say sorry, because they may be very too confidential, right? So in this manufacturing process, we start with the cell bank. And in this case, we use the HEK cell, which is the human embryonic kidney cell. And this also, the HEK that we use, there are different cell lines. What we use is 293. And this is owned by someone. We have to pay heavy money to buy this from them. Because these are so expensive, we try to make a master cell bank. From there, we try to make thousands of working cell bank. And as you know, the microorganisms, so these are human cells though, but just like microorganisms, you can go up to certain passages. As you know that once you keep on treating the microbes, they mutate, right? So as the passage going on, they mutate, they change their characteristics. So that's why there are some regulations put around that you can passage it so many times, so that the inherent characteristics can stay as it is. So we start with a small vial, as I say. This may be a 0.1 ml vial, which is the cell bank. We take it, we start expansion, we put it in the media where the cell is going to grow, and then we do multiple passages so that we are not shocking the cells. And we grow it, and once we have certain volume, we transfer it to the bioreactor. And the bioreactors are different in different industries. When I was in the vaccine industry, we had like huge 1,000 liters, 2,000 liter bioreactors, stainless steel. It's like, you know, I don't know how many have you seen the beer manufacturing, you know, those big drums where you're doing the fermentation is the same thing. We are doing fermentation in these bioreactors. We are making lots and lots of cells. In this one, we came up with a very interesting bioreactor that is called isolus. We also call it a donut. It is not a typical bioreactor that you see in the industry, those steel tanks. These are glass bioreactors. Very efficient, and because we need to be very careful about this manufacturing process, we also need visibility that how the cells are growing, and this really helps a lot. So in the manufacturing, again, I'm going to focus on microbiology because the microbiologists are always watching this. they're looking at the environment, and I'm going to touch it in the next slides, that what they're looking at. They're looking at the process. They're looking at the critical process parameters, that these bioreactors are such that if one bug goes in somehow, it can start propagating very fast if it is a fast grower. If it is a slow grower, obviously it takes time, but it grows, and a microbiologist can say from their growth that, hey, my bioreactor is contaminated, and in that case we have to dump the entire run, and this costs millions of dollars. So that's why it is very important to control the entire process and the environment, and microbiologists plays a great role in this, because they can say where the leak can happen. They can say that where you are not doing the setup right, and they can say that your interaction with the process is not right, and that's how they can control the environment of this manufacturing process. So the cell growth. Okay, there's a spelling mistake. It says call growth. I'm sorry for that. It should be cell. Poor eyesight. Then the transfection happens. So we use plasmids in this cases, and there are different 3 types of plasmids we use that we then inject to these bioreactors, and the virus, which is our product here, that is made. In the gene therapy, again, as I said, this is a very unique sector now, because you are relying on a virus to take this particular gene that the patient is lacking, injecting to the cell, and then trying to make that particular protein in your body that you're lacking. So also in the industry, when you will be coming, you will be hearing a lot of terms: orphan drugs, rare disease, and we deal with orphan drugs and ultra rare diseases. The drug that I'm working on is called DMD. There were some movies done in India on DMD. So this is a particular huge protein that lacks dystrophin. The protein is called dystrophin, that is this kids cannot manufacture, and this is X-linked gene. So most of the patients are generally male, means boys. The girls are not much impacted, because, as you know, there are XX versus XY. So if your X gene is defective, the boys are mostly get impacted by this genetic disorder, and they cannot make this particular protein, and they lose their mobility and their muscle functions, and this is a fatal disease. So that means that at certain point they die. Most of the kids die within the age of 14 years. Some kids we have seen can live 20, 22 years also. And what we are doing right now, we are trying to make this drug that is injected in the body, the protein is made, and they get their mobility back, muscular function back. The dystrophin protein is very, very important for this kind of functions. Okay, going back to the manufacturing again. So we make this virus, and then now we have this virus within the cell. I said that we started with the human cell, right? So inside these human cells, the viruses are there. We want to break down the cells. That is called the cell lysis, and then we need to harvest the product. And so also you will hear the term upstream versus downstream. So all this are called upstream process, means in the manufacturing, these are the upstream. And then comes the downstream process. Downstream is all about purification. As I said in the beginning of my discussion, that it is very important that the drug is safe. Safety is not only from microbiological perspective, it is also from other contaminants, the byproduct that the cells are making. So we do lots of purification. There are so many steps in the purifications that we do different types of purifications, and it will take, again, another whole class to explain these purifications. So we do purification, 1 step, 2nd step, 3rd steps, 4 steps, and after that we do the nanofiltration or the viral filtration, because this is very interesting. When I came, I said, okay, when your drug is a virus, right, you're trying to make, and the virus that we use is the AAV virus. So we said, when you're making a virus, how you're going to filter out the virus? And this is where all the brain comes in, right? A microbiologist or virologist comes in. Man, I'm trying to make a virus, and how I'm going to filter out the other viruses or other organisms that can be present. So you have to understand the size of the virus, molecular weight of the viruses to kind of design this filtration process. Again, microbiologists. So that's why. And one more thing I would say that here, when we see the people coming in from the industry, again, our teachers were great. They taught us the basics of microbiology, and I see lack of that knowledge. So, okay, if I keep on talking, I'll go in a different direction. So let me come back to this. So we make this, we do the filtration, we make first the bulk drug substance. And again, we are making so much of the drug at a time, we cannot filter everything. So we make this bulk. It is, in many cases, these are sterile drug substance bulk. So they are in a big bag. We put it in the -80 freezer so that we can store them for the long time. And when it comes to the time comes to make the fill, means the actual vials, we take it out, we thaw it, and then we do the formulation. The formulation buffers, there are different types of buffers we use to do the formulation. It is generally to make the stability, to make the drug stable. And then we do the final formulation, and then we do sterile filtration. Again, microbiology is coming into place. Here we use the 0.22 micron filters. In some cases, for the larger molecules, we have to use 0.45 micron filters so that we can filter out the organisms. So after the filtration, then they goes inside the vials that you see in the market. The doctors take it out and use so that they put the injection and take out the drug and inject you, right? So that is what we call the final product. It goes inside those vials. And now in this case, I went over very fast, but again, there are a lot of things in microbiology comes into play. When you are manufacturing a product, as I said, they are very prone to microbial contamination. And in the industry, we are very, very concerned about gram-negative bacteria. And the reason we are interested in that, because as you know, when the gram-negative bacteria breaks, their cell breaks, because their cell contains LPS, lipopolysaccharide, we call them endotoxin. And this can cause severe fever and in some cases shock and death of the patient if there is high concentration present. So in every step of this process, in certain place, we take the drug and do the in-process testing. We check for microorganisms, we check for endotoxin, okay? Our ultimate goal, again, as I said, to make a safe drug so that you know that the drug that I'm giving to my son or my friends or families, it is not going to cause any harm, right? Okay. The filtration also, again, the microbiologist is involved in this process because you know that bacteria or other microorganisms at their different growth phase can change the structure and also the size, right? So as they're growing in their stationary phase, it can be a different size versus in the growth phase, it can be different size. Okay, so that's why the filtration steps is important. And after that, we do check again for bacterial growth or endotoxin. I will see if this is very interesting. I just went very fast, but again, how the plasmids are introduced here and how the virus is made is a very interesting genetic engineering that happens in this place, but anyhow. Okay, so once the drug is made, it goes to the lab for testing, rigorous testing. There are so many different types of tests we do. But before even it comes to the test, as I said, we take samples from the process. So when the process is happening from the bioreactor, from the purifications, we take a sample and we test. Again, the purpose of the test is to make sure that you don't want to test the drug at the end because you may have lost control in that time. So you are testing it in different phases so that you can say that ultimately your drug is safe. And right now in the industry, there is a term going on, risk-based approach. It means we are designing so that we can take away the dependency on the end product testing. You are designing risk-based design. You are trying to figure out where your weaknesses are in the process, and you can put the mitigation in place so that ultimately you don't have to test so many times, right? And FDA has adopted and EU has adopted. So ICH has come up with ICH Q9 guidelines, sorry, Q10, to talk about the risk-based approaches. I get confused with those numbers most of the time because there are so many ICH guidelines. Okay, so in process samplings, as I said, from the bulk drug substance, as I said, that we make, we take that sample. We also take during the drug product, and obviously the finished product we test. And the testing involves chemicals and microbiological testing. So chemical, they check for identity, purity, strength, and microbiologically, we are looking for anything that can cause patient safety, right? So we are looking for any kind of bugs that could be present. We are looking for endotoxin, and there are some other tests that we do, especially for the biologics, which is mycoplasma and adventitious agents. Means we want to make sure that there is no other types of virus present in the drug. So we do all those testings. Now that is the quality control, and we hire a lot of microbiologists in the quality control lab, especially for the microbiological testing. We also hire microbiologists because so as I said, it is a risk-based approach. Now, all the regulators are saying that, guys, look, you cannot depend on the end product testing. Hire microbiologists and people who can design your process from the very beginning. When you're making a building, hire a microbiologist to do the design with you, because they can say that what kind of paint you should use, how your HVAC system needs to be. Because if there is a high moisture, microbiologists will tell you, guys, the mold is going to grow, right? If they say that, hey, if you do not put the 0.22 micron filters or 0.45 micron filters, the air that is coming into your clean room, okay, clean room is the term that we use in the industry where we make the drug, right? It is called, it is started in the term started in the electronics sector, where they were used to make the chips, because the chips cannot have a single dust. It is going to screw up its function. So they came up with the different types of classification, ISO, and they call ISO 5 class, 6 class, 7, 8, and the pharmaceutical industries, the biotech adopted those classifications. It means the purity of the air. In the air, how many particles can be present, right? And then Europe and FD or US harmonized together to come with a classification. They call it now grade A, B, C, D. Grade A is the purest air. Again, the microbiologists, they go in, as I'm saying here, the environmental monitoring, this part. So we do air and surface monitoring. So the microbiologists, they have different equipment. They take it there, they test the air. They say, okay, I have this many particles present, and does the guideline allows that I can have it? And once they make sure that it is, then they said, yes, it is the safe environment to manufacture your drug. Grade A is the highest purity air. We don't allow any microbes to be present there. We collect a cubic meter of sample, air sample. I believe when we studied the aeromicrobiology, we did some of this in the field, collected different samples. So just like that, again, things are getting very sophisticated nowadays. All are those petri dishes and things are going on. But Europe is little traditional still. They still want us to put some petri dishes in those environments to make sure that the heavier particles or microbes are settling down on those plates, and we can see some growth on those plates. But right now, most of the air monitorings are equipment based. So we are collecting, and then these are going to a plate, and we are hoping that because of the stickiness of the plate, the microorganisms are going to stick there. So the air passes on those plates and they get to stick there. Okay, so going back again to the design phase, contamination control strategy, heavily focused on this term now, contamination control. And the whole Annex 1 has been revised, and lots of emphasis has been put on contamination control. So contamination control means they are saying, design the facility so that the bugs cannot enter to your environment and the process. As you know, it is almost impossible, right? Because we carry billions of bugs on our on our skin, and we are the people who are going inside the clean room. So right now, it is a well-known fact that in a clean room, human beings are the most are causing the most contamination in those clean rooms. We shred, I don't know, millions of cells every minute or every hour, something, those crazy numbers. So in this clean room, that's why we came up with certain types of gowning. And this is also based on those electronic industry experience. It is a full body gown. Nothing is exposed. No skin is exposed. And then these people are working in those places so that we are not contaminating the process. So Ashwini, please check the time and let me know, okay, if I'm taking too much time, please.
You can continue, sir. No problem.
Okay, thank you. So during the manufacturing process, the environmental monitoring is happening, as I said, and the surface monitoring, we use a plate called RODAC. And these are, again, sticky plates that we touch different surfaces, including the operators who are working in the industry, and take the sample and do microbiological analysis to see what kind of organisms are present. Again, as I said, we are very curious about gram-negative bacteria, and obviously we don't want any mold, because if you have a mold in a clean room, it is very, 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 very difficult to get rid of mold. So those are in the field, in the manufacturing area. In the lab, then we bring these plates, we do identification of the microbes. Again, microbiologist comes in play, because no one else has any clue what those things that are going on the plate, what they are. Only the microbiologist can do the identification. So that's why we need microbiologists there. I touched on the endotoxin testing. We are very, very interested in the endotoxin testing. There are certain limits put on every drug based on the volume. We check for the endotoxin to make sure that, again, it is safe. When I started in 1999 in the industry, or before that, there was a rabbit pyrogenic test. We used to inject the rabbit with the drug to see if it is developing temperature, but those are gone. I don't know if anyone does it anymore. Endotoxin is now the replacement. We do microbial count of the in-process and the drug substance samples. Again, as I said, we take different samples. So the microbiologists do filtration method. So we used, again, there are a lot of sophisticated filtration equipments that are found. We do filtration method. We do spread plate. We do pipette techniques, like in some cases where the drug product is very viscous or cannot go through the filters, we use this kind of techniques to make sure that the drug is, again, free of microorganisms. And ultimately, for the injectable or the pan products, we do sterility testing, and the purpose is that there cannot be any organism present. Okay. Then comes my part, where I moved in right now, the quality assurance part. But before that, there is regulation. As I said, every country has different regulations, and we always struggle with this, because in many cases they are not harmonized. In India, you have the Central Drugs Standard Control Organization. I browsed in the internet and figured it out, because I had no idea. If I'm wrong, please excuse me. I hope this is the organization who controls the drugs. Obviously, FDA is very famous. Obviously, EMEA for the Europe. Japan has their regulatory agency. China. So it makes it very, very difficult for us when we are going to different market to meet those regulatory expectations, because everyone has different expectations, and the regulation is different. It is the biggest struggle. That's why to go to different market. Recently, there was a family reached out to me from India, and their son was diagnosed with DMD, and their family said that we need help. Can you include our child in the clinical trial? It is not that easy because of the regulation. We don't have any clinical studies, so it saddened my heart, but I had to say, I'm really sorry, we cannot do it, because again, the regulators doesn't allow us to go in a market just like that and start the clinical trial. So based on the like we cried a lot from the industry, and that's why at certain point some of the countries came together and they created this ICH. Some of the countries harmonized, and that's why it's called International Council of Harmonizations. Not all the countries came, China and all, they are still observing, but there are many European countries and USA came together, and there obviously then there is who. We then comes at the end, before the drug goes to the market, then we come into the picture. We make sure that the drug met the quality that it was intended for. When in the industry, we say that if you don't write, it never happened. That's where the batch record comes in. Whatever those manufacturing people and everyone is doing, every time they need to record it in a batch record. The main reason is that, again, I need to go back and make sure that you're supposed to mix it for 2 minutes, 10 seconds. You mix it for 2 minutes, 10 seconds, and not 2 minutes, 30 seconds. If it is 2 minutes, 30 seconds, we call it a deviation. It is a deviation from from your standard operating procedure, and we have to understand the impact of the deviation. Because these are so precisely calibrated, all these steps, you cannot do whatever you want. The drug will be adulterated. So quality assurance people, so we have people who check this, every step, every entry. There is a SOP, a standard operating procedures. We have for everything, we have a procedure written so that people can follow that procedure. There are work instructions written that you have to do this microbiology things that they do. For everything, there is a work instructions. You have to take this much, you have to do like this, you have to put on your gloves. The gloves should be put on this way. Every details has been written. And many of these are written by a microbiologist, again, to make sure that people are not doing something wrong. Then we check the test results to make sure that this is met against the specification. A certificate of analysis is generated. And from there, we generate a certificate of conformance, and the regulators wants it, and then we release it to the market. So that is the quality assurance function. So why microbiology is important? In this industry, as I said, aseptic operation, right? Sterile drug manufacturing. You will see you can find it in the YouTube. At some point, operators used to smoke in the clean room. They used to like scratch their head and start operating. Every time you are touching a surface, and this is where every bugs are sitting, right? So every time you're touching a surface, you are contaminating your hand, and then it is going into the process. What you are seeing in this picture is just one of the examples. We take fingerprint of every operator that are working in those aseptic core to make sure that there is nothing growing in this hand. And there is, again, IPA, 70% isopropyl alcohol present or ethanol present, so that they are sanitizing their hand all the time. We sanitize all the surfaces, disinfect the wall, top to bottom, the ceiling, everything, every day, okay? So why you need to wash your hands? A microbiologist teaches them why this is important. Why gowning is important. As I said, your body is continuously shredding skins and germs in the environment. So that's why this particular type of gowning is important, that your skin is not exposed to the environment. Why you cannot be sick and work in the clean room? Because as you're coughing, you are generating those droplets, right? And those can go to the product, right? You can contaminate the product. So that's why a microbiologist can tell them the importance of all of this, and why and what microbes are important in the clean room. What can be present? Only a microbiologist can make a good assessment of it. We in the industry are very, very, as I said, curious about bacteria and fungi only. Other microbes, we know that they cannot survive in this particular controlled environment, so we are not very much interested finding those. And why they cannot be present in the clean room, as I said, because if it's present in the clean room, you know that somehow they are going to land up to your process or to your drug. Now, that being said, again, coming back to this, you can go anywhere you want after you're graduated. You can pursue a doctorate degree and become a teacher. You can go to the industry and work, go to the become a soil microbiologist, and there are so many different sectors. The reason I chose this path, obviously, it was highly rewarding. As I said, ultimately there are a lot of recognitions there. You are being proud that you are making a life-saving drug, and a patient can take the drug, and you know that you're going to save someone. So you have that pride when you do it. As I worked in the rare disease industry, I've seen many times we bring in the patients so that they can speak directly to the manufacturing operators and tell them the importance. So that's why I've chose this industry, obviously recognition, and obviously Duddu, right? Duddu is very important. Money is very important. This is an industry where you get paid a lot. If you can make out, you can figure out to make products. Again, lots of Duddus, man. There are billions of dollars flying around, money's flying around, recognition. So that's why I think this is a very important sector for us. Okay, I believe that is the last slide, Ashwini. Yeah, and then thank you.
Thank you, Vishwarup sir, for very informational and wonderful lecture. It was truly informative and very helpful, particularly for life science students from microbiology background. There are a few questions from the audience, which I want to hand it over to Dr. Jamuna. And before that, I have one personal question. Sir, while pursuing masters in microbiology, so in between the semesters, our students, they do get the ample time gaps. So if someone have aspired a career in pharma sector, what are the gaps in terms of training and exposure they can prepare themselves with that so that they can be ready for the industry?
Thanks, Ashwini. That is actually a great one. I don't believe, sir. Please remind me. I don't believe we had those that time when we were in masters. I don't know whether we were allowed to go or we have those semesters. It was a 2 years degree, right?
Yeah.
Yeah. So here, this is happening now. What we do, we bring in students during their break. This is called the summer program, so that they can get exposure to this industry. You can learn so much in a college setup, but when you see it, this is the practical experience that you're gathering. Then you know, again, I told you so many things about clean room, the process, the vessels. Unless you see it, you really don't understand that how these are designed and why these are important. So I will say that, yes, if there is opportunity exists, if you can work, again, we do accept students. There are students coming in, we accept them. We cannot take him to the core. We show them from the outside. We have the viewing gallery that we use for the regulators. So bring them in. We also allow students to work in the lab. So they can come and work for 6 months during the 3 months during the summer break, and they can do the hands-on experience. So that is possible. That again helps you to translate that you're studying to your practical experience. Did I answer your question, Ashwini?
Yes, thank you.
Sure.
Over to you. Good morning, sir. I'm Dr. Jamuna.
Please don't call me sir, okay? Every time you guys are calling me sir, it is making me really embarrassed. So please, you call me Vishwarup, okay?
Okay. So I'll be moderating the Q&A session. First of all, I would like to tell you guys that career choice can be a real dilemma for the graduate students. And also, it is an unusual time and situation as we're all dealing with both the professional as well as the personal consequences of a global pandemic. So this talk definitely helps licensed students to decide and understand the various options and avenues for pursuing a career in life science as well as the biotech sector. And I have collated a few questions from the audience, so I'll be directly to you. The first question is from one of our audience. It is post-COVID-19 situation, all population are getting more and more aware of what a devastation a single nano monster can bring in. Do you think microbiologists can play a bigger role in future, especially in pharma, biotech, and clinical sectors?
Oh my God, it is absolutely, it is absolutely, trust me, the questions. So if you see people, there are two types of people in the US. Uh, Again, I'm not going to go into the politics, but some people who have no understanding and they are followers of certain individuals, they're doing things, crazy things, right? And if I want to explain here, it will become all political discussion, so I'm not going there. So when I interact with such people, or when we are talking about a vaccine, and when we see these non-believers, they believe that this is all a hoax and made thing up, they don't look at the data. The microbiologists can help them to explain the data, that look at this, how the virus functions. And I won't say that I was successful every time, but in certain cases when we talk to them about how a virus functions and how it is different from your typical flu that you're seeing, some of them got it. Obviously there are people who never want to believe you, and they will never believe you. They will think that these all are made up. So so when you understand the basics, say that, okay, I know that how this functions, this can then come into play in developing the drugs, going to the clinical trials. And that's why, as I said, we hire microbiologists heavily in these industries because of this basic understanding. If you know how a molecule is designed, how a virus or an organism is designed, right? You can develop those particular drugs so that it can be targeted drugs. And for that, the basic understanding is important. So that's why I believe personally that in the industry, we need more microbiology, especially in this case of situations where you do not have much time to react. You need this kind of people who understand the organism and know what exactly to target on.
The next question is, how do industry and academic CVs differ? So what should be included in the CV when it comes to applying to an industry? And how to structure a CV?
Okay. So the CVs are very interesting thing. I have done CVs back in Bangladesh. I've seen those CVs, and we do CVs here too. If you see my CV, I hope you guys have seen it. It is. I even chopped it down more, because here, when you're coming from the industry, I need to focus on what I learned when I'm coming to the industry, I need to focus on what I learned in my school. And here it is very important not to write, but how you applied it. And I think going back to Ashwini's first question, that how can you boost your experience, right? So I will say that whenever you're giving an opportunity, you're getting those break, whichever sector you want to go, whether it is in the biotech or anywhere, try to find a link, and your teachers can obviously help you to make those connections, to make those visits, and put it on your CV that I have observed this, okay? I have worked there for 3 months. This obviously helps when the resume comes to us. We do look at that. We also look at, recently I hired someone who has experience, who have educational background but have no experience. She worked in a daycare. The reason I hired is that, again, it is showing me, when I asked her that why you want to come here, sometimes the personal touch. She said that I have seen kids who are suffering, and then I thought that, hey, I have knowledge in microbiology. Why don't I go and see if I can do some contribution to make this life-saving drugs? So sometimes it depends on the person who's getting your CV. I know in India, again, to be very blunt and honest, you need to have a lot of connections to get a job and do certain things. Here it is a little different in many cases. So it depends on how you're presenting yourself. Try to always when you are being interviewed, try to make a connection with your experience, with your educational background, with what you're trying to do. and why this is important for you that you are applying for this job. Read the job description very well. Okay, don't just go there and say that I want to work here. Understand the job description, read every line. When a manager is putting a job description, he or she is thinking what he or she is looking for. So, and then write your resume accordingly. That's how we tell people that try to make the CV relevant to the job description so that it is, I'm not throwing it away on the first look.
There is another question from Dr. Nagendra Prasad. He wants to know, with vast industry experience you have in your company, you hire people from all the nukes of the globe. What do you think are the strengths of the students that are coming in from Indian universities in comparison to students coming from other countries? And what are the immediate fortifications our students do need to stay ahead?
This guy asked too many questions, Nagi. You have to go. You have to go one by one, okay? He's my best buddy, though. We spent so much good times there with Srini, Raghavendra. We used to go. There was a near. I used to stay in the hostel there, boys. So there was a big tree, oak tree or something. We used to play cricket there. Sometimes we used to call bunking the class. So maybe sometimes you don't notice. We used to like kind of take the back door and we used to go out to play cricket. But anyhow, Nagi was our leader to take us out there. Okay. Go one by one of those questions. What is his first question?
The first question is, what do you think are the strengths of students coming from Indian universities in comparison to students coming from other countries?
Okay. Obviously, I believe I touched on it, the statistical part. We see a lot of people are coming with IT background, with statistical analysis, and then those are the two sectors right now we are seeing heavy industry, heavily populated with Indian population. IT sectors, and then maths, statistician. There are obviously certain number of students we get from Harvard and MIT in biologics, but most of those are, very few are Indian. Most of them are from either China, Japan, or those descendants. So that is my experience with Indian students.
And the second question that he has asked is, what are the immediate fortifications our students do need to stay ahead?
What are the immediate what?
Fortifications.
Certification?
Fortification.
Oh, fortification. Sorry. Okay. Sorry. Sometimes, these things are not very good. Sometimes the network sometimes really disturbing. So I don't know, honestly, some of the things I cannot, I myself can fortify. This industry, this is a place where most of the time
Yes, sir, we are hearing you. You have muted. Please unmute yourself.
There's something signals coming up. Hold on. Hold on. Let me reboot. 1 second, please. Can you hear me now?
Yes, sir, we can hear. Yeah.
Okay. It could be the network. Sorry. Go back to the question again.
It was, what are the immediate fortifications our students do need to stay ahead?
So I will say that again, the basic knowledge, as I said, was very, very important, right? To have that basic knowledge of microbiology. The statistical part, have your strengths on maths, try to analyze the data. We have to do a lot of data analysis. So that is also very important that you do data analysis. At our time, I don't know whether we did a lot of those analyses. We just looked at the data, but statistics is playing a very, very key role to, and because in the clinical trials, the data analysis is very important. So for the Indian students' perspective, that is also important.
Okay, the next question is, being a faculty, we would like to know how to train the students for the industrial career. What needs to be included in the curriculum?
Visits, trips to the sectors. Okay, so try to make those as many times as possible, because you cannot make the connection just by reading the books. It is, you have to see it. Once you see it, you know what it is, right? You can ask questions. Bring some industry experts in during your sessions so that they can do some presentations, and it also depends on the interest. Like medical microbiology, for example, I don't know if you can get some doctors or someone who come really from those sectors and do the presentation, who does those kind of analysis. So that helps again to comprehend this particular knowledge.
Okay, the last question is, after graduating, is there any certification or crash course for students to join pharma companies?
In US, there are. I don't know in India if there is any, but in US, we do have some places where they do. I won't say crash course. There are like different length of courses they do, 6 months to 1 year. That gives you the perspective of basic knowledge in the industry, in the biotech industry. But again, I'm not sure whether those things are available in India. I'll say that honestly, you don't need it. If you can make those trip to the industry, and as you said, if there is a break between your semesters, try to use those time to go and work with someone. We say job shadow. So that really is important. Then you can talk smartly during your first interview.
One more question just came up. Is it necessary to have prior knowledge of and training in pharmacovigilance as well as regulatory affairs?
It depends on which pharmacovigilance and all. Again, we don't expect when you're coming out of college, right? We don't expect that you know everything. That's why we train people. We have robust training. There's a training departments where like people are working to design this to train you so that you can learn. But as I said, the most important part is to have that basic knowledge. Okay, if you have the basic knowledge, if we see that you are not just coming here to waste time, so the interview is very important. We do check a lot of things. Again, I cannot give you everything away in US training part, but we have certain things that we are trained on during the interview process to look at. If I feel that you're faking, you're done. I'm not hiring you. So as I said, I hired a person who had no industry experience working in a daycare, but I felt that she is a really genuine candidate because she has that honesty in her mind to say that no, I want to make some changes. I want to do something good for the people.
Thank you, Mr. Vishwaroop, for answering all our questions.
Absolutely.
One small question from my side, Mr. Roop.
Okay, sir.
Okay, I just want to know, in this era of a lot of artificial intelligence getting integrated into various curricula, how much of AI can we incorporate in our curriculum to see that our students have an edge over others?
So thank you so much for asking that. I was expecting someone to ask that. This is booming. This is another booming part here. And that's why I was saying that the statistics, the math is important. You know the biology. You know the biology. You know the basic thing of microbiology, and then you know the computational part of that, right? And you're putting this together to come up with certain things. We get so many equipment in the lab which are designed by microbiologists or of artificial intelligence, and this is good and bad in one sense, because the more we are buying this, we are hiring less because the machine can do the job. But at the same time, this is opening up another sector where the microbiologists are working with those computer centers to come up with this kind of technologies that can do the analysis. And the good part of it is that always when human are working, they are prone of errors, right? We always make mistakes, but the machine doesn't. So here there is a booming sector where they are trying to come up with again, the microbiologists and the computer scientists are working together to come up with a fully automated system that can do the analysis and no human, like right now, as I was saying, I'm doing the filtration part, right? So those are not needed. We call live online process testing. So as the drug is being manufactured, this particular machine is doing the detection whether the microorganism is present there or not. So I'll say that this is going on great, sir, honestly. And if people can think, not that is another sector. Thanks for bringing that up. If they can start thinking about it and start making those connections and gain some knowledge in those. Again, I don't understand computer very well. My son has to come and fix my computer most of the time. So if they can gain that knowledge of the software and the hardware and how this comes together, then it is great. It is great. That is another sector. Obviously, that is a very another sector that is developing right now. Actually, have been developed. I shouldn't say developing. These are developed, but these are again getting better and better every day.
Thank you very much, Mysuru, for the very good and informative lecture. And it has opened up many vistas probably for our students, and probably to encourage them to look for various other avenues they would think of. At the outset, I would like to thank you for the wonderful lecture. I would like to just mention at this point of time that our Vice Chancellor, Dr. Surendra Singh, is a former Director of National Institute of Biology, and he is well known in the area of vaccine production. And similarly, our Pro-Chancellor, Suresh, as you know, he is the President of the Pharmacy Council of India. And we in JSSAHER, we are at a very good ecosystem, I would say, wherein a medical college, this is primarily a health science-based university with a lot of understanding of our life science students, with medical college students and pharmacy students. So there is a good interaction that is taking place. Recently, we are also launching on trying to introduce life skill sets. Particularly, we have in India life science LSS, LSSDC, which means Life Science Skill Set Development Corporation concept, which has identified very clear areas for life science students to see that their skill sets are improved. This is one good development that has taken place. And we in our department are trying to incorporate all these things. And we also, as you rightly said, summer internship is a must in our curriculum, unlike the ones that you studied and I was teaching there. The summer internship was not a must. We have summer internship, which is a must for our PG students. So also project work at the final year. So there is good hands-on training that we give in our system. That's what helped us to attain good ranking systems in the international level also. I wish your visit to our institute would be of great value, both for you and also for us, because we will be actually mingling with the pharmacy masters and the vaccination masters or biologic masters, both of whom are responsible for heading all this. And the Dean Faculty of Life Sciences wanted to be here on this day. Unfortunately, but because of some hard engagements, they had to leave the Mysuru. Anyway, whenever you visit us, we would be able to interact with you. He is Dr. Bala Subramaniam, who really laid the foundation for our Faculty of Life Sciences. Thank you very much for your wonderful presentation, and we look forward to interacting with you and collaborating with you in future for any of our requirements. And you are also welcome for any of your collaborative or research or other results. Thank you once again. It was nice seeing you after such a long time. I could not remember your face. And Raghavendra told me, no, Raghavendra did his PhD with me only. And I told him that you are coming. That he said, sir, he is your student. I never knew until that. And thanks once again for your informative talk. Thank you.
It's an honor, sir. Thank you so much for giving me the opportunity. Obviously, Ashwini made it happen. It's nice to see you. And obviously, whenever things settle down, this COVID thing scares goes away. Obviously, plan to visit with the family.
Please do. Please do. You're welcome.
Thank you, Vishnu sir, for giving a wonderful lecture, and you were so enthusiastic in answering all the questions even at this time over there. Thank you so much. Now I request Dr. Sumana, Assistant Professor, Department of Microbiology, to propose our reports.
Thanks, Dr. Ashwini. I offer my humble pranams to the lotus feet of His Holiness Jagadguru Dr. Shri Shivaratri Rajendra Mahaswamiji and Dr. Shri Shivaratri Deshikendra Mahaswamiji. As rightly said by Eckhart Tolle, acknowledging the good is the foundation for all abundance. It's my pleasure to propose the vote of thanks on the occasion of 105th Jayanti celebrations of Dr. Shri Shivaratri Rajendra Mahaswamiji, commemorating International Microorganism Day. I owe my thanks to our chief guest and speaker, Mr. Mysuru Dasgupta, Director, Quality Assurance, Gene Therapy, for his insightful talk on career opportunities for life science graduates in Pharma Biotech sectors. Thank you, sir, for introducing new avenues available in pharma companies where life science students pave their career into it. I express my profound gratitude to Pro-Chancellor Dr. B. Suresh, Vice Chancellor Dr. Surinder Singh, Registrar Dr. B. Manjunatha, Director Academics Dr. P. A. Kushalappa, Dean and Director Research Dr. S. Balasubramanian, Professor and Head Dr. Ravi Shah, for their unconditional support, guidance, encouragement, and inspiring mentorship. Thank you all, sirs. My sincere thanks to Dr. Ravindra, Chief Information Officer, and team for all the technical support and logistics for the smooth and successful conduction of this event. Thank you all, sirs. My cordial thanks to Dr. Ashwini and Dr. Jambana and all the faculties of the department for the cooperation. My deep sense of appreciation to all the faculties, non-teaching staff, and all the participants for being a part of this event and acknowledging it. Finally, I extend my thanks to all who extended their favor directly and indirectly for successful conduction of this event. Thank you all. Sarve Jana Sukhina Bhavantu. Stay safe and healthy and have a nice day.
Thanks, everyone.
Thank you.
Take care. Bye.